Hey everybody, Dr. Phil from medpathway.com. Today's question that we're looking at is plasmid DNA and relative electrophoretic mobilities with varying forms of the plasmid. So you have supercoiled plasma, you have nicked plasmid, and you have linearized DNA. So imagine that you ran each different type in an agarose gel, and then after electrophoresis you ask which one is which. What is the relative mobility differences between the three forms? And so think about how gel electrophoresis of DNA works. Most DNA, especially for plasmas, will be done in agarose gels. Smaller fragments might be done in acrylamide gels. Now, if you think of the DNA chain as a long ladder and you're carrying it on your shoulders through the forest, where the forest is basically tantamount to the matrix that the agarose gel forms, you can see that shorter fragments are easier to carry through that particular forest, whereas the longer ones are going to be more impeded, and so you know that smaller fragments migrate faster. So what about the electrophoretic forms of the plasmid DNA? So you have supercoiled, as you can see on the left. As you digest it, you can relax it by nicking it, and then if you completely digest it, you can linearize it. So then how do the supercoiled and relaxed forms compare in their mobilities to the linear form? Well, if you take a look here, what you can see is the pattern. What you see is that the supercoiled form is the most condensed form, and so it can go easier through that matrix, through those trees in that forest, which is the analogy for the agarose gel, whereas the relaxed form, the nicked form, is got broad shoulders, it's bulky, and it's having trouble getting through, whereas the linear form is intermediate in its mobility. So that's how you run the electrophoretic mobility of the three forms of a plasmid DNA. Come check us out at medpathway.com.